Okay, hello, this is James Lewis. I'm the Medical Director for the North Carolina Division of Public Health Surveillance for Healthcare Associated and Resistant Pathogens Patient Safety Program, or NCDPH SHARPS for short. Just a quick disclaimer, we were having some technical difficulties with the recording the first time around, so I'm re-recording these first several slides. So if it sounds a little different than you remember, if there's anybody who is listening to this again, I apologize, or if there's a little bit of not quite seamless transition between this re-recording and then the subsequent full presentation recording after these first several slides. Apologize for that. So we're going to talk to you today about CRE surveillance, identification, containment, and response. And I'm going to be presenting with three of my other SHARPS team members. And so the SHARPS team here is Jennifer McFarquhar, our program director, myself, the medical director. We have three epidemiologists, Heather DeBendris and Katie Palladino, formerly Katie Steiter, recently married. They will be helping present with Jennifer and I. And then we also have a newly hired, just started on Monday, health educator and campaigns coordinator, Deborah Dolan. We're very excited that she has joined us. And then we also have Savannah Carrico, another one of our epidemiologists who started around the same time as me last fall of 2017. And then hopefully coming soon, we will have an epidemiology program manager. Unfortunately, soon may be a relative term, but we do hope to fill that position sometime in the near future. So our objectives today for this presentation are to discuss the public health significance of CRE and the process for surveillance and detection, as well as discussing a unified response to CRE and describing containment strategies for CRE in individual facilities. So at the end of the presentation, as you probably gathered from our pre-test questions, we would like for infection preventionists to be able to identify resources and key stakeholders related to MDRO investigations, as well as assess appropriate infection prevention measures and to initiate a effective investigation. We would like for them to understand their role in the control of CRE, as well as other significant multi-drug resistant organisms in North Carolina, and improve early recognition and control of transmission of these organisms. And this will require a coordinated rapid response and effective communication related to MDRO, specifically CRE for this presentation. So just a quick disclosure, all of us, all of the presenters for this presentation today have no financial conflicts of interest to disclose. And then we're going to skip over these questions as the presentation's over and we're not getting active responses currently. So now let's just talk quickly about what a multi-drug resistant organism is. So CRE is what we're here to talk about today, and CRE falls under the broader umbrella of multi-drug resistant organisms. And this is a nice little cartoon here. So you are the next class of drug-resistant bacteria. As humans continue to abuse and overuse antibiotics, your ranks will swell. So go out there and mutate. And remember, that which does not kill us makes us stronger, which is definitely true with regard to bacteria. So what are multi-drug resistant organisms? So they are germs that have become resistant to several kinds of drugs that are normally used to treat them and can cause infection in any part of the body. So any of these bacteria can cause infections in any part of the body. Also fungi. And because multi-drug resistant organisms can be spread from person to person, there is the opportunity for spread within and between facilities by the transfer of patients either infected or colonized with a multi-drug resistant organism. Colonized individuals can spread multi-drug resistant organisms to others, even if they are not symptomatic or even if they are not actively infected. And so they would not necessarily be aware that they are colonized as well. And vulnerable patients may be more likely to contract a multi-drug resistant organism because they are more prone to infection due to either increased age or perhaps additional comorbidities like specific diseases such as diabetes or compromised immune systems due to things like cancer or HIV. And they also may have increased exposure to healthcare in general, as well as invasive devices, which would increase their risk for acquisition of multi-drug resistant organisms. And then resistance to drugs can make infections with these organisms difficult to treat, which could increase associated morbidity, mortality, and cost of care. For example, the mortality rates for CRE that invades into the bloodstream can be up to 50%. And that's a significant rate of mortality for that type of infection. And then multi-drug resistant organisms in general, so CRE plus other types, which we'll talk about on the next slide, cause 2 million infections a year in the United States annually, approximately 2 million infections a year, and cause approximately 23,000 deaths of those 22 million infections. And so we'll skip over this answer again, but here you can see that a lot of people thought it was 250,000, but it's actually 2 million infections a year. So what else is an MDRO besides CRE? And probably the most common one that everybody is familiar with from the news is MRSA or MRSA, and that stands for methicillin-resistant Staphylococcus aureus. Other ones that people probably are very familiar with in the healthcare industry are VRE or vancomycin-resistant enterococci. A lot of places screen for that. And then also C. diff or Clostridium difficile, which is a bacteria that causes diarrhea in patients who have been exposed to antibiotics generally. That's who's at highest risk for acquiring C. diff diarrhea. But other types of bacteria, so those were gram-positive, and then C. diff is a gram-positive rod, VRE and MRSA are gram-positive cocci, but CRE, MDR Acinetobacter, MDR Pseudomonas, and ESBLs are your gram-negative multi-drug resistant organisms. And while we're primarily talking about CRE today, ESBLs are also important, and that stands for extended spectrum beta-lactamase producers. And then multi-drug resistant Acinetobacter and multi-drug resistant Pseudomonas are very important. They are environmental gram-negatives rather than Enterobacteriaceae, which we'll define on the next slide. And those are important because they can harbor carbapentamases, just like CRE can, which is a very important mode of potential transmission. So let's talk more about CRE. So CRE stands for carbapenem-resistant Enterobacteriaceae, and Enterobacteriaceae are a gram-negative group of bacteria that can be found in the digestive tract of many mammals. And the two most common carbapenem-resistant Enterobacteriaceae that cause infections in people or colonize people are E. coli or Escherichia coli and Klebsiella species. So this is any Klebsiella species. So the most common one would be Klebsiella pneumoniae, but also relatively common is Klebsiella oxytosa. There are many other types of Klebsiella species, and all of them can be considered CRE, and as you'll learn later, can all be reportable, or are reportable in North Carolina, nationally notifiable as well. But Enterobacteriaceae is a broad term that encompasses more than 60 genera. So the genera is the Escherichia here, or the Klebsiella here. Also Enterobacter is another genera. And each of those have multiple species under them. So you know there's over 60 genera, and then within each of those genera are multiple species. So you can see that there are a lot of different species of Enterobacteriaceae, and they can all become carbapenem resistant. And CRE is Enterobacteriaceae who become resistant to carbapenem antibiotics. So what are carbapenems? They're a class of beta-lactam antibiotics, kind of like penicillin, but they're an advanced penicillin. And there are 4 different carbapenems available currently. So there's ertapenem, meropenem, imipenem, and doripenem. You may see sometimes that they're combined with other drugs. Imipenem is always, or almost always, combined with cilastatin, and then meropenem, there's a recent formulation that just came out called meropenem with Vabor-Bactam that you may see as well. But these are the 4 carbapenem antibiotics that are available: ertapenem, meropenem, imipenem, and doripenem. That's not to say that there may be a new one that comes out eventually, but right now, these are the 4 that are available. And these drugs are usually reserved to treat very drug-resistant infections, so infections with bacteria that are resistant to multiple types of antibiotics. So all CRE, while important, and we want to respond appropriately and get people on appropriate precautions, usually contact precautions for any CRE, of particular public health concern are carbapenemase producing CRE. And that's also referred to as CpCRE. So a carbapenemase, what is that? This is an enzyme that a bacteria can synthesize or make that can break down carbapenem antibiotics and essentially inactivate them and make them unable to kill the bacteria that they are targeting or that's causing infection. And there are multiple different types of carbapenemases, and that gives us an opportunity to come up with a lot of very complicated names and acronyms to simplify those names. So we'll go through a few of the most common ones here. So first up is Klebsiella pneumoniae carbapenemase, then the New Delhi metallo-beta-lactamase, and then Verona integron-encoded metallo-beta-lactamase, or VIM, imipenemase metallo-beta-lactamase, or IMP, and oxysolinase 48 or OXA48. And these are particularly important because they can be encoded on mobile resistant elements called plasmids, which are pieces of DNA that reside in a bacteria that the bacteria can use to make these enzymes. And what's particularly concerning about them is those pieces of DNA can be transmitted from one bacteria to another that's completely unrelated. So normally when you think about bacterial resistance, you think about a parent bacteria transmitting a resistance mechanism to its offspring. But with plasmids, you can have a plasmid transmitted between just two neighbor bacteria, even though they're completely unrelated, which means it's a much more efficient way to spread resistance. And that's illustrated here by this nice little cartoon. So this is a nice infographic from the CDC about CRE. So this came out in 2013, but unfortunately things have not gotten any better since then. CRE is still considered an urgent threat in the United States. About 9,000 or around 9,300 drug-resistant or CRE infections are caused per year, and of those 9,300, about 600 deaths. And those are primarily due to, or the 9,300 that are estimated are Klebsiella species, so Oxytosa or pneumoniae, and then E. coli. Now there are other types of Enterobacteriaceae like Enterobacter that also cause CRE infections, but these two are the most common and is what this particular infographic is referring to. So now we'll move on to what DPH is doing. We've kind of established that this is a significant threat and requires a response. So what is the Division of Public Health doing to help mitigate this? So we have 4 specific areas that we work under. So first is detection. So we want to increase awareness. We perform sentinel surveillance and testing at the state lab, as well as colonization screening. And then we also want to ensure rapid response and containment, which we'll get to a little bit later. We're not going to talk today about stewardship efforts and education, but these are also very important parts, and part of education is giving this webinar. So other forms of education are also important, and we participate in those as well, but today's example is the webinar. So we'll move on now to talking about detection of MDROs. And first we'll talk about detection and surveillance. Right.
Yeah. Yay.